In this video, we will be having a walkthrough, or basically a rundown, over uh, all flashcards that I have created in preparation for an exam that I took a few months back. This is in preparation for the grade 12 fourth quarter biology exam. So yeah, let's proceed. So okay, if by the way, if you're not familiar with how Anki works, in terms of the way I use it, in several cards, I use something called image occlusion enhanced. Basically, you place boxes such as these to cover certain words, and those words are what you intend to memorize. So here, the one in red is what we have to um, find out. Basically, for archaebacteria, they have less standard energy production. And at the same time, they live in extreme environments, and their cell walls are made of different matter. So compare that with eubacteria. Um, so if archaebacteria have less standard energy production, for eubacteria it's the opposite. They have more standard energy production. So knowing that archaebacteria, for archaebacterium, uh, for archaebacteria, cell walls are made of different matter. For eubacteria, the same components are composed of peptoglycan. I'm not sure if this term is synonymous with peptidoglycan, uh, but just to be safe, just note this term. At the same time, eubacteria cannot survive in extreme environments, so it's the opposite for archaebacteria. Now, going to what they have in common. So both have no nucleus and organelles. They are both single-celled, and they're prokaryotes. And they evolved from a common ancestor. Okay? Alright, let's go to the two main categories of bacterial infections. We have gram-positive. So, in terms of physical appearance, like specifically their color, the bacteria is usually blue or purple um, if they're supposed to be classified as gram-positive. If the bacteria is seen to be pink or red, then it's gram-negative. For gram-positive bacteria, basically the peptidoglycan is thick and has many layers. On the contrary, for gram-negative, the peptidoglycan is thin and is composed of only a few layers. Okay, process of gram staining. Um, gram staining, by the way, is, I believe, the method that is utilized in order to determine um, if a sample of a bacteria is either positive or negative, gram positive or gram negative. So yeah, what was stated earlier is also stated here. Deep purple or blue is gram positive. Pink or red is gram negative. What is the science behind the color? Basically, for gram positive, the cell wall absorbs the crystal violet. Crystal violet, this is basically one of the um, materials, substances used in the process of gram staining. At one point, for sure, you apply crystal violet in the petri dish, I think. I'm not sure if a different instrument is used, but I think petri dish is what is used because we're still dealing with bacteria technically. So yeah. Um, for gram-negative bacteria, the cell wall does not absorb the crystal violet, but instead it absorbs the safranin. Safranin, I believe, is applied prior to the application of crystal violet. I may be wrong. Again, this has been a lesson a few months back. I can't remember everything. But yeah, gram-negative bacteria have a hard protective outer shell. And yes, this explains why uh, gram-negative bacteria are more difficult to treat. That's because of their harder cell wall. So if your infection is determined to be composed or be if your infection is discovered to be caused by gram-negative bacteria, then definitely it would be harder to treat it, uh, especially with medicine. Okay, seeds versus spores. For seeds, they include flowers and gymnosperms. Um, for spores, mosses and ferns have them. Seeds do not need water. They can live anywhere, and they are protected by a seed coat. And yeah, they provide a food source for the embryo, and they can disperse in many different ways. This is why, well, technically gymnosperms are classified uh, in terms of having seeds. For a fact, I think I've mentioned this in a previous vid. Gymnosperms are the most diverse plant species uh, because of many factors. More diverse uh, compared to its other counterpart. Um, I can't really remember, but basically, larger trees classify under that category. Okay, spores. We have mosses and ferns. They have no protection from harsh conditions. New plants must make its own food and they must grow where it lands. They need something for production and they must live near uh, water environments. Water, yeah. They need what? Water for reproduction. I'm not sure if, well, okay, I'm already sure about what I'm gonna say. Certain particular plants, uh, I think mosses are one of them. They do not grow as big as other plant species for the reason that they do not have a proper or a complete vascular system uh, that is supposed to be responsible for the management of water, taking water in and taking water out through respiration. Yeah, aerobic respiration. Anyways, moving on. Structures found in bacteria. The cell walls of bacteria are made up of what? Glycoprotein murin. And yeah, the main function of the cell wall is it helps in providing support, mechanical strength, and rigidity to the cell. And for number three, it protects cells from bursting in a hypotonic medium. Hypotonic, I believe, if I recall correctly, hypotonic. So hypo means low. Uh, Hypotonic, low in the context of salt concentration. So the tendency is if there is no cell wall in a hypotonic environment, the cell would lyse or burst because of the significant imbalance in the proportion of salt to water. Basically, an imbalance in solute to solvent. Okay. Uh, okay, we've been here already. Cell walls are made of different matter, while for eubacteria, cell walls are made of peptoglycan. Structure relevance and the significance of diatoms to the environment. Uh, yeah. For the structure, they are unicellular with a silica-based cell wall. So what's their relevance and significance? So basically, they contribute significantly in the removal of CO2 or carbon dioxide from the atmosphere through the process of photosynthesis, converting it into oxygen. So this is what diatoms look like. And they are a primary food source for higher organisms in the food chain, and they play important roles in energy and nutrient cycles of water resources. Okay, Archaebacteria live in extreme environments. For eubacteria, they live in normal environments. They cannot live in extremes. Structures found in bacteria. For the ribosome, number one, basically ribosomes are a tiny granule made up of RNA and proteins. They are the site of what? Protein? Is it protein? Yeah, protein synthesis. And for the third characteristic, they are free floating structures that help in transferring the genetic code. So in this image, where do we find the ribosomes? Over there. So the free floating structures, you can see them uh, described as in terms of dots, red dots. Okay. Fungi. Main structure, they consist of long thread-like structures, hyphae. There you go. These hyphae constitute a mycelium. Uh, when you hear the word mycelium, that's automatically fungi. And they're unicellular. What is the function of mycelium in the first place? How's it relevant? What's the point of knowing that? Part of a typical fungus. So in nutrient transportation, they help provide a transportation network to pass nutrients along the fungal body. In the context of reproduction, mycelia of two compatible fungi can fuse together, allowing cells to combine and for DNA to mix. So in a certain way, maybe don't quote me on this, but I personally think that mycelia might be just synonymous to dating apps. If that helps you remember a definition that's closest to the function of mycelia in the context of reproduction, then it may be useful, right? Who knows? But of course, find whatever is useful to you. Yeah, that's all I'm gonna say. Pheromones allow hyphae of two distinct mycelia to follow each other as they grow. Pheromones cause haploid nuclei from each parent to exchange genes. Okay, so yeah, pheromones allow the hyphae of two distinct mycelia to follow each other as they grow. Alternation of generation. In non-vascular plants, the dominant generation is the gametophyte. In vascular plants, the dominant generation is the sporophyte. Okay. So, coming from the gametophyte, let's start here. This is a cycle, by the way. So, I'll just choose. I can just choose wherever I want to start. So we'll start here. So with the gametophyte, the gametes would eventually fuse, and they would form a zygote. So male and female gametophyte, they form a zygote. And after that, the zygote undergoes mitosis, so it becomes a sporophyte. The sporophyte would undergo meiosis, so the result would be 4 spores, and one of them would undergo mitosis. Well, not just one of them. Everyone will have their own mitosis, but in this illustration, only one of them is shown to proceed to the gametophyte. I believe that's already self-explanatory that the remaining 3 would also undergo the same process. But yeah, I may be wrong, but how could it be wrong, right? Anyways, sporophytes are diploid. So as you can see here, 2n. 2n means diploid. Single n means haploid. Okay. Non-vascular plants, they are plants that do not have a vascular system. They're often small. I think three plants are classified under this category: liverworts, hornworts, and mosses. I hope I remember my biology right. Okay. We've seen that already. Spores versus seeds. Okay, I'm not... You know what? Fine. Developed by mitosis from the ovules. So, spores are produced by meiosis of the sporophyte. For seeds, they are developed by mitosis from the ovules with fertilized egg cells. They are produced in fewer numbers, or seeds. Spores, on the contrary, are produced in large numbers. Spores require more water for germination. For seeds, they require less water. Okay? Produced by blank of the sporophyte. Okay. Meiosis, spores. Mitosis, seeds. I think that's one way to remember that arrangement. Okay, bacteria is blue or purple if it's gram-positive. So just to give you an illustration of what was being described earlier. So the peptidoglycan is thick in gram-positive bacteria. So as you can see here, the purple lines, they constitute, well, you can say that the layer is thick. And on the contrary, for gram-negative, the peptidoglycan layer is thin. There are just a few layers. But what makes gram-negative bacteria more difficult to treat or more difficult to kill? I believe it has something to do with the outer membrane. So it's harder to pass through this particular layer. Okay. So, the use of fungi as recyclers, they break down large carbon molecules in organic material. They return essential building blocks to the soil. These nutrients are made available to plants again. Fungi are decomposers, they break down plant and animal debris. So, fungi have many uses, basically. More water, spores, seeds, less water. Units of asexual for spores. For seeds, units of sexual reproduction. Structures found in bacteria. For the cytoplasm, what describes the cytoplasm? They basically help in cellular growth, metabolism, and replication. The cytoplasm is a storehouse of all of the what? All of the organelles. Just kidding. Chemicals and components to sustain a bacterium's life. Okay. So, what are the chemical processes that occur within a cell? ATP biosynthesis, transcription, DNA synthesis, DNA replication, I mean, not synthesis. Okay, we've been there. Okay, what are the prokaryotic modes of nutrition? So based on carbon source and energy source that can be used by prokaryotic organisms to synthesize organic compounds. Basically, so we'll start here. So prokaryotes, it has two types, autotrophs and heterotrophs. How are they different? They're different in terms of their carbon source. For autotrophs, they have carbon dioxide as their carbon source. For heterotrophs, they have organic compounds as their carbon source. For autotrophs, we have photoauto, and chemoauto. Uh, Basically, these two have in common the characteristic of having CO2 as the main carbon source, perhaps the sole carbon source. But for photo, from the word photo, um, well, citing the word photosynthesis, light is used as an energy source. For chemo, chemicals are used as the energy source. For heterotrophs, there are also two types, photohetero and chemohetero. Light is used as energy source for photohetero, but what makes it different from photoauto is that um, it manifests the characteristic of heterotrophs. That is, they have organic compounds as a carbon source. Same goes with chemoheterotrophs. Um, it's the same with chemoautotrophs, except that um, it manifests the characteristic um, of using organic compounds as a carbon source. So that's it. It's just a matter of memorization. Just don't get confused. Okay. Structures found in bacteria. It is also known as a cytoplasmic membrane or cell membrane. Yeah, in other words, the plasma membrane could be called the cytoplasmic or cell membrane. Number two, it is composed of phospholipids. This is a kind of lipid. Proteins, carbohydrates, forming a what? Uh, a layer. Fluid mosaic. Fluid mosaic. Number three, it helps in the transportation of substances, including the removal of wastes from the body. And it helps in providing a mechanical barrier to the cell. So, yeah. I believe lysosomes are basically the garbage bins within a particular cell. And I don't know if lysosomes uh, willingly expel themselves from the cell together with the trash, but essentially wastes are removed. Gates are opened for waste removal thanks to to our plasma membrane. And yeah, mechanical barrier. Um, we release waste products, but at the same time, uh, we don't just let anyone inside the cell coming from outside. So the plasma membrane is responsible for that. They are a house gate in a way. Fifth, plasma membrane acts as a semi-permeable membrane. Like what I said, it doesn't accept everyone from outside, um, and it doesn't really. It's not designed to let everyone from inside exit. So, yeah, it only allows selected material to move inside and outside the cell. That's what I just said. Okay, how many cards do we still have? I don't really know. Can't really tell. Um, Fungi, mycorrhizae. Fungi that have a symbiotic relationship with the roots of many plants. Majority of plants in this world are able to actually grow better in terms of their ability to extend their roots thanks to fungi. So as you can see here in this image, for plants that are not associated in any way with mycorrhizae, uh, they have shorter roots. With mycorrhizae, evidently, you can see that the roots are more extended. So yeah, at the same time, um, Yeah, like what I said, fungi have a symbiotic relationship with the roots of many plants. What do fungi get in return for helping plant roots? I know there's an answer for that. I'm not sure why it's not here. But basically, just to add one more characteristic that describes how mycorrhizae help plant roots to be specific. Um, so mycorrhizal fungi form with plants common mycorrhizal networks or CMNs to signal nearby plants in response to herbivore and pathogen infestation. So in a way, they who is that man who is he John Deere? The famous guy who said um, the British are coming. Basically, he was able to warn um, locals of an upcoming on the upcoming arrival of British colonial forces. We're talking about America before um, it officially gained its independence from Great Britain. Anyways, basically, m- common mycorrhizal networks are designed to signal nearby plants in response to something that may um, destroy plants, basically. So, herbivores, animals, or other organisms that feed on plants, leaves, and yeah, pathogen infestation. Okay, protists. How do ciliates perform sexual reproduction? Uh, so occurs through conjugation. Conjugation is the exchange and recombination of nuclear material between two compatible ciliates. Okay, this is what is the singular form for ciliate? Let's check. Cilium. I think it's cilium. Is it? I think it's cilium. Anyways, kingdom protista. So for the Rhizarian supergroup, we have foraminiferans and radiolarians. Where do foraminiferans live? Bottom ocean and freshwater. For radiolarians, they live in oceans and warm surface waters. Slime molds are heterotrophs. Okay, symmetries. No symmetry, one example is sponges or porifera. Radial symmetry, nidaria or what is this? Sea anemone? I'm not sure. Bilateral symmetry, example, arthropoda or arthropoda. Whoops. Radial symmetry, an example is a starfish. I think it's echinodermata. Bilateral symmetry, a fly. This is arthropoda. What is the impact of prokaryotes on humans? They help digest food. They produce crucial nutrients. They protect from pathogenic microbes and they help train the immune system to function correctly. Okay, we've been there already. Structures found in bacteria. The plasmid. Plasmids are a small circle of DNA. They are bacterial. Well, bacterial cells have many plasmids. Plasmids are used to exchange DNA between bacterial cells. Okay. Basic types under kingdom Animalia. We have mammals, birds, reptiles, amphibians, invertebrates, fishes, and yeah, that's it. Just take note of those characteristics. Why are sponges classified as animals? They are multicellular, heterotrophic, they lack cell walls, and they produce sperm cells. So yeah, sponges also reproduce sexually when specialized gametocyte cells produce sperm and eggs. Okay, we've been there. Foraminiferans are eaten by marine animals. Radiolarians, small creatures, to be more specific. So, what is the importance of gram staining in medicine? Basically, gram staining is a lab test that checks for bacteria at site of suspected infection or certain bodily fluids. Basically, a med lab scientist processes the gram stain, giving quick results. And from there, healthcare providers can know if bacteria are present and identify the general types. In other words, gram staining is one of the most common ways to quickly diagnose bacterial infection in the body. So, I believe from there also, um, the healthcare provider is able to identify which antibiotics are appropriate for uh, the infection that you may have, depending on whether it is caused by gram positive or gram negative bacteria. Because again, they are both different in terms of characteristics, uh, more notably in how easy or how challenging it would um, kill them, knowing that gram negative bacteria are more difficult to kill, like what was said earlier. So capsule is a kind of slime layer which covers the outside of the cell wall. They are composed of a thick polysaccharide. It is used to stick cells together and works as a food reserve, and it protects the cell from dryness and from chemicals. So where is the capsule here? It's over here. Okay, we've been there. Been there. Non-vascular plants. So there are no roots, stems, and leaves. The gametophyte again is dominant. What's one example? We have bryophytes. So a more specific example of bryophytes would be mosses. They have no true roots. Instead, they have a rhizoid, a root-like structure. On the contrary, for vascular plants, they have roots, stems, and leaves, and the sporophyte is dominant. We have three under this category: pterophytes, gymnosperms, and angiosperms. Pterophytes, they have spores instead of seeds. Under this category, we have ferns, horsetails, club mosses, gymnosperms, and angiosperms. Uh, Seeds are involved in the continuation of their species. Gymnosperms, conifers, angiosperms. This is one confusing idea because I remember the largest tree in the world. I think that's the Sherman something. It's located in the U.S. Uh, Technically, large trees are classified as gymnosperms. Maybe there's a distinction between deciduous trees and other trees. I hope so. Or things would be more confusing if it was otherwise. Okay, applications of gram staining. Earlier, we have identified the importance in the medical field. So it is used to test for bacterial or fungal infection. So this is the counting of bacteria, selection of suitable culture media, for microbiotests, and screening the quality of clinical specimens for etiology and explained and unexplained infectious cases. After all, we don't know all diseases yet. We haven't discovered everything yet. So, what's the main basis for classifying plants? One is whether they have spores or they have seeds. Yeah, whether they reproduce by spores or seeds. Gymnosperms and angiosperms have seeds, as said earlier. Ferns, club mosses, and horsetails have none. Also, another basis is the presence of vascular tissues. Okay, I've already mentioned this earlier. Let's move on. Main basis for classifying fungi, morphology of the mycelium, its ability to reproduce sexually, asexually, or a combination of both, and their mode of spore formation, as well as the type of their fruiting bodies. Okay, four types of extremophiles. All of them resist the denaturing effect of their respective environments. So for thermophiles, from the prefix thermo, they can withstand hot temperatures. They have blank, also useful for biodiesels. They have enzymes, heat stable DNA polymerases, that are useful for biodiesels. We have psychrophiles. This is for the opposite. They can live in cold temperatures. So antifreeze protein to protect DNA, for example, protease. Halophiles, they live in extremely salty environments. They have adapted by having reduced solubility or osmotic balance or acidic proteins that can resist denaturation by salt. The salt removed from cytoplasm is triggered by the use of energy. So one example is Halobacterium halobium. Number 4, acidophiles. They can live in high acidic environments. Efficient mechanisms to keep their cytoplasm at neutral pH. Some have acidified cytoplasm. One example is Helicobacter pylori. This is pretty famous, by the way. Uh, if you ask me its actual or other name, I don't know. Well, I could just say that it's famous because I've seen it many times. So it's only a matter of great familiarity. Need water for reproduction. Flagella. So what's the role of a flagellum? So this is a rigid rotating tail. It helps the cell move clockwise and anti-clockwise forward, and also helps the cell spin. The rotation is powered by a hydrogen gradient across the cell membrane. So pili are receptors that attach to surfaces for reproduction. The hairy stuff. They're receptors. And yeah, they are short protein appendages. They are smaller, clearly, they are smaller than the flagella. They fix bacteria to surfaces so that they would attach. In a way, they could be described as adhesives. And for number 4, they help in the reproduction during conjugation. Okay, 8 main characteristics of animals. First, they are multicellular. Second, they have a eukaryotic cell structure. Third, they have specialized tissues, connective, muscle, or epithelial, or maybe all of them. They are able to reproduce sexually, and at one point in their stage of development, uh, a particular stage is described to have a blastula. Number 6, motility or movement. Number 7, heterotrophy. Number 8, they have advanced nervous systems. And yeah, what is the meaning of heterotrophy? Ability to ingest food. Make up 90%? No. Ocean sediment or floor. Okay. Okay, what are the uses of fungi in medicine? So they could be used as cholesterol biosynthesis inhibitors. Usually, if you have high cholesterol, you need to take in certain antibiotics that help solve the issue of having too much cholesterol. So, cholesterol biosynthesis inhibitors uh, are actually made out of fungi, and it's effective, that's for sure. Number 2, medicinal mushrooms for lung diseases and cancer. Although, when we say cancer, um, I believe this information has been affirmed, but I'm not confident that it can be the sole solution to solving cancer cases. Okay, number 3, penicillin. It is intended to treat bacterial infection. Antibiotic or antibacterial agents. They can be used as antifungals, antivirals, and immunosuppressants. Okay, plants. Seeds versus spores. For spores, they are unicellular and they are the first cell of a gametophyte. For seeds, they contain within them a developing embryo. So when we say embryo, that's already associated with seeds. Okay, I think that's all for this Anki deck. More videos coming soon if I have time. I'll see you soon. Thanks for watching.